just so that I don't forget, because I did that on a seminar once and had to ask the speaker to re-record their session. I managed to record the Q&A bit, which was obviously, that's the more organic portion. But yes, thank you everyone for joining us today, and welcome to this STEM Village Immunology seminar series event. My name is Katie. I'm stepping in for our usual host, Matthew, as he's busy dealing with family stuff at the moment. So I'm very pleased to have the opportunity to meet our speakers today and hear their presentations, and hopefully have you all engaging as well. I should say my pronouns are she, her. And I'm going to hand you over quite rapidly to our first speaker. A short introduction, first of all. So our first speaker is Joe Rouse. He is joining us today from Wisconsin, and having started his academic career at the University of Wisconsin-Parkside after his tour of duty in the US Army. His interest was sparked by host microbe interactions, and received his BS and MS doing research in Dr. Greg Richards' lab, studying metabolic stress response in E. coli. He was inspired by professors that had a positive impact and wanted to pursue research further, and so is now on the PhD research program at the Medical College of Wisconsin, and is in the middle of his PhD work in Dr. Rob Lockhead's lab, studying immune dysregulation in Lyme arthritis. So I will mute myself and hand you over to Joe. Thank you, Joe.
Thank you. Thank you for the introduction. Let me go ahead and share my screen. Okay, so yes, thank you, Katie, for that introduction. My name's Joe, and I am coming to you live from Milwaukee, Wisconsin in the US. And today, I am just going to talk a little bit about my research in Dr. Rob Lockhead's lab at the Medical College of Wisconsin in the Department of Microbiology and Immunology. We primarily study Lyme disease, and we have a little social media page, which is Lyme MCW. And so the major point of that, just a little plug for that, is that we hope to share accurate and evidence-based information about Lyme disease, because just based on historical records, there isn't a lot of accurate and evidence-based information out there. So we're trying to help that with that social media presence. So give that, check that out if you have time. But today I'm going to talk about my project. Again, I am a third year in my PhD program, so I don't know, for us, it's probably about halfway through. So I'm just going to walk you through my project, which is sort of investigating the role of peptidoglycan in Lyme arthritis development. And so just a little bit of information about Lyme disease in general. It's caused by a tick-borne spirochete, Borrelia burgdorferi. This is a diderm bacterium. Most refer to it as a gram-negative bacteria. And it is transmitted by ticks of the Ixodes genus. And you can see it's sort of transmission tick cycle here in this graphic by the CDC. And humans are in fact accidental hosts. This is the most common vector-borne disease in North America. It's quite common in Europe too, but it's just the most common here in the US. And here is a sort of cartoon pictographic of what a Borrelia burgdorferi might look like. And then this is sort of a scanning electron micrograph image of that as well. And you can see sort of that the incidence of Lyme disease and as well as the geographic location has increased in the last few years. And you can see that in this graphic from the CDC, going from 2001 all the way up until about 2018, you can see the reported cases by county in the US has spread quite a bit in the last just 10 years. So Lyme disease can come with a variety of symptoms based on the stages of infection. Usually the earlier stages come with the characteristic erythema migrans rash, which looks like a bullseye, although only about 75 to 80% of patients get this rash, so it's not always the case. Further development of symptoms can include neurological symptoms such as facial palsy, and then eventually joint pain and swelling, multi-joint pain and swelling, I should say. Lyme arthritis is the predominant late-stage manifestation of the disease, and most patients can resolve this by just appropriate antibiotic treatment. However, in about 10 to 20% of patients, Lyme arthritis can persist even after months to years, even after antibiotic treatment. And so this is kind of where our research comes in as to what is the deal with this persistence. One of the features of Lyme arthritis is that synovial lesions can develop, and they look like this. And synovial lesions actually in Lyme arthritis are similar to those in other forms of arthritis, such as rheumatoid arthritis. And so it turns out that a lot of what we've been learning about Lyme arthritis over the last few years or decades has similarities in rheumatoid arthritis as well. One of the key things that has been sort of revealed about Lyme arthritis in humans is sort of the more cellular mechanistic causes or features. And one of the main features is about Lyme arthritis is that it is accompanied by this dysregulated autoimmune like CD4 T cell response and a significant interferon gamma response. And this is sort of signature of Lyme arthritis. And so what you see here is evidence from my PI in the past showing that, or which cells are involved and that these T cells, so you can see sort of the range. This is some flow cytometry data and scatter plots, and you can see that there's a large percentage of T cells that are involved, and a large percentage of those T cells are in fact CD4 T cells and some CD8 T cells as well. And so he goes on to show that these T cells are interferon gamma positive. And so, like I said, a sort of signature of Lyme arthritis is this robust interferon gamma response, and the cells responsible for that response happen to be CD4 T cells. So we see a large Th1 response in Lyme arthritis. And so sort of the major questions that the lab is investigating is what perpetuates this Th1 interferon gamma intensive response. Recently, it's been shown that peptidoglycan is a persistent antigen found in Lyme arthritis patients. It's been shown that peptidoglycan from Borrelia burgdorferi was detected in the synovial fluid of patients with Lyme arthritis after even antibiotic therapy. And so you can see in this graph here that they were able to detect Borrelia peptidoglycan in the synovial fluid of patients with Lyme arthritis. And it says N equals 34, so there were about 34 subjects in this particular study. And I think all but maybe 4 or 5 of them had been treated with both oral and IV antibiotics, and peptidoglycan was still seen in synovial fluid. The interesting thing about this is that throughout these patients' experience post-infection, they haven't been able to detect Borrelia burgdorferi in these patients. And so despite the fact that they're not able to detect by PCR or by culture the presence of active bacterial infection, they're still seeing Borrelia peptidoglycan present. This same study from Dr. Brandon Dutres also continued to show that Borrelia peptidoglycan can induce arthritis in BALB/c mice. And so peptidoglycan has actually been used in murine studies to induce arthritis for many years. But this kind of focuses on peptidoglycan from Borrelia burgdorferi, specifically being able to induce arthritis in mice. And you can see in these pictures taken swollen joint spaces, and then you can see in this graph of measured arthritis, and they usually just measure the area that they show here, and the percentage of arthritis goes up with injection of peptidoglycan from Borrelia burgdorferi. And so this is sort of, like I said, even though peptidoglycan has been associated with arthritis in both disease cases and lab research, this is sort of novel in that it's actually peptidoglycan from Borrelia burgdorferi, so it's in the context of Lyme disease. And so my particular niche of the project, my little vein of the project, is sort of focusing on what's the role of peptidoglycan in Lyme arthritis pathogenesis. And so just a little bit of background information on bacterial peptidoglycans. It's a essential cell wall component in both gram-negative and gram-positive bacteria. If you zoom in on the structure, it looks like this. It's made up of repeating subunits of N-acetylglucosamine and N-acetylmuramic acid with this peptide link. And there's a couple unique features about Borrelia peptidoglycan in that it is shed upon replication rather than recycled. So most bacteria have a recycling system, but Borrelia doesn't. It's just shed in copious amounts upon replication. And another thing is that its structure is a bit different. And so you can see that one of the key features of this peptide link is DAP, diaminopimealic acid. And actually Borrelia peptidoglycan doesn't have that. Instead, they have L-ornithine in place of that. And so their structure is a bit unique. And so it's easily identifiable as Borrelia peptidoglycan with its unique structure. So peptidoglycan in general is recognized by certain pattern recognition receptors, such as TLR2, which is more of a surface pattern recognition receptor, and NOD2, which is known as sort of a cytosolic pattern recognition receptor, or within the cell. And because of this, there is sort of conflicting data in the literature about pattern recognition or receptor recognition of peptidoglycan and how it influences inflammation in general, or the immune response. And so you might think intuitively that TLR2 activation leads to NF-kappa B signaling and inflammation, pro-inflammatory cytokine and other pro-inflammatory mediator production. NOD2 has been shown to have some roles in mitigating sort of overactive TLR2 pro-inflammatory signaling, but NOD2 itself can also induce NF-kappa B signaling. It's just sort of the appropriate regulation of NF-kappa B downstream signaling as to whether you have an anti-inflammatory response or a pro-inflammatory response. And so that's why there's sort of a gap in the knowledge with this particular way of immune induction. And so this sort of brings me to the hypothesis of my project, which is that peptidoglycan from Borrelia burgdorferi can act as sort of an adjuvant by activating different types of these innate antigen presenting cells that might detect peptidoglycan through these pattern recognition receptors to elicit this pro-inflammatory response, which brings in the CD4 T cells, which I showed earlier. There's evidence that those are sort of the major sources of interferon gamma and this interferon gamma response that's seen in Lyme arthritis patients. And so the hypothesis really of my project is that that is how this interferon gamma response is perpetuated by this lingering peptidoglycan acting as sort of an adjuvant for antigen presentation or antigen presenting cells to activate its immune response. And so the first sort of goal of my project was to determine the effects of peptidoglycan on different types of these innate antigen presenting cells, such as sort of classical antigen presenting cells like macrophages or dendritic cells, as they are known to express these pattern recognition receptors. And then more particularly to investigate whether or not it's both of these or one of these receptors that's involved in the signaling, and whether or not these cells exhibit a sort of pro-inflammatory phenotype. The second part then is to determine the T cell response. And so if these sort of activated antigen presenting cells can induce this T cell response, and then to actually investigate in vivo peptidoglycan's ability to act as an adjuvant. And so just as a sort of alternative approach, I just wanted to introduce the fact that there is evidence that interferon gamma has effect on not just those characteristic or classic innate antigen presenting cells, but also other cell types in the area. So in like the joint space, such as synovial fibroblasts, which I abbreviate as FLS, And there is evidence that shows that interferon gamma, a high interferon gamma environment can actually prime the synovial fibroblasts to be immune effector cells as well, similar to what we see in more classical ones such as macrophages. There's just evidence to show that in patients being stimulated in ex vivo with both Borrelia burgdorferi and interferon gamma has a lot of transcriptional effects on synovial fibroblasts. And we see upregulation of certain things like I mentioned previously, such as pattern recognition receptors, particularly those that I mentioned before, NOD2 and TLR2, as well as other genes involved in just pro-inflammatory induction and even antigen presentation, such as MHC class 2 molecules. So I just wanted to throw that in there as sort of a alternative interest for another cell type. The two models that I use in the lab mostly are wild type black 6 mice that sort of have a mild response to Lyme arthritis, and they maintain a strong tissue repair response, compared to a black 6 IL-10 knockout model that exhibits severe Lyme arthritis, similarly to what we see in humans, driven by dysregulated Th1 and interferon gamma responses. And again, these have sort of an impaired tissue repair response. So those are the two models that I focus on using in the lab. So to get at this first question about what sort of effects peptidoglycan has on these different cell types, and looking for a cell type specific effect of or response to peptidoglycan, I set up an experiment that sort of looks like this. So basically I took bone marrow derived macrophages from both of those models that I just mentioned, black 6 mice and the IL-10 knockout mice. And I stimulated bone marrow derived macrophages with peptidoglycan and interferon gamma together, or of course alone, versus unstimulated for control. And I stimulated them for about 24 hours, and then did flow cytometry to analyze the surface markers expression to sort of get to their phenotype, whether they were looking like more immune effector cells or not. And I also collected the supernatant from the culture to look at cytokine and chemokine production. And so for macrophages, what we saw was actually with interferon gamma, a sort of increase in MHC class 2 molecules on the surface. Like I said, this is flow cytometry, so we're looking for surface marker expression. And we saw a trend upward of MHC class 2 molecules. And again, if actually I stimulate these cells for longer than 24 hours, we would definitely see an increase as well. And so that was to be expected compared to unstimulated macrophages. However, what's interesting is when we stimulate with peptidoglycan and with peptidoglycan and interferon gamma together, MHC2 expression decreased quite significantly. Now that coupled with the fact that expression of co-stimulatory molecules CD80 and CD86, and so these, if you're not familiar, these molecules, CD80 and CD86, are co-stimulatory molecules, and so they're known to provide sort of a secondary signal for T-cell interaction. And what's interesting is that when stimulated with interferon gamma and peptidoglycan together, there's a dramatic increase in the expression of these co-stimulatory molecules on macrophages. And so in this case, even though CD80 and CD86 are known for co-stimulation, the thing is that what they interact with on the T cell, they can also be immune regulatory. And so when you, in the absence of signal 1, which would be more of an MHC to TCR interaction, you have this signal 2, which is this co-stimulatory molecule interaction with the T cell, that in the absence of the signal 1, this would provide more of an immune regulatory signal so that T cells aren't just activated unnecessarily. And so this would sort of suggest, again, that a classical APC like a macrophage would be interacting with T cells less with MHC2, as we see up here, and more with these co-expression of these co-stimulatory molecules. And so that suggests more of a tolerizing effect on macrophages in response to peptidoglycan. And this experiment was done in the wild type black 6 mice. And it was also done in the IL-10 knockout mice, but there wasn't a significant difference between the two. So I just decided to show the wild type. Now, as I introduced that alternate cell type, synovial fibroblasts, we are interested in those again because they can sort of, there's evidence to show that they adapt this sort of immune effector phenotype in this high interferon gamma environment. And so I did the same experiment that I just explained, but with synovial fibroblasts rather than macrophages. And what we saw here was actually when stimulated with peptidoglycan and with peptidoglycan interferon gamma together, an increase in expression of MHC class 2 molecules. And that's interesting because it was quite the opposite of what we saw in macrophages. And it shows that it's actually sort of interferon gamma independent. And so in response to being stimulated with peptidoglycan, fibroblasts adopted this sort of MHC class 2 high phenotype, which suggests, again, the ability to present antigen, which normally these synovial fibroblasts wouldn't be doing that. As another marker that we looked at, IL-15 receptor, I was interested in looking at IL-15 receptor expression on fibroblasts to see if there was any other sort of immune interaction other than just these cells interacting possibly with T cells. And so IL-15 receptor expression sort of suggests a more innate response, so a interaction with natural killer cells or NK cells, because that's mediated by IL-15 and IL-15 receptor. And so we saw that when stimulated both with peptidoglycan and interferon gamma together, there was a increase in the expression of IL-15 receptor on these synovial fibroblasts, which might suggest that they not only have the ability to interact with other T cells by antigen presentation via MHC2, but also that they may interact with NK cells via IL-15. And so this sort of further characterizes that previous evidence that these synovial fibroblasts might be acting as sort of immune effector molecules in this disease. And so basically, I was able to sort of start with addressing the very first part of the project that I mentioned. My future directions mainly are to do that second aim as well, to look at the T cell responses. But I just wanted to point out that these cells were stimulated. I did mention they were stimulated with peptidoglycan, but we use sort of a commercial peptidoglycan. And so we'll have to do these experiments again with peptidoglycan derived actually from Borrelia burgdorferi. And then again, look at those cell-specific responses. Because we have seen some cell-specific responses, I definitely want to continue and see if these responses were TLR2 or NA2 dependent by using knockout mouse models and doing the same experiments. And then going on to continue that T cell response, and then looking at peptidoglycan's actual ability to act as an adjuvant in vivo using a transgenic mouse model that sort of controls for antigen presentation. As we've seen, there's evidence in literature that shows that some mouse models, such as the IL-10 mouse model that I was using, can have sort of a non-specific T cell response, so that can obscure some of the data. And so we plan on using this transgenic mouse model that controls for non-specific T cell activation to look at peptidoglycan's role as an actual adjuvant in vivo. And so I won't take up any more of the time, and I can ask any questions if you guys have. I just wanted to show a little picture of what our lab looks like. This is myself, my PI, Dr. Rob Lockhead, and then other members of the lab, Becky and Amanda. Let's do a little plug for NCW.
Thank you. I always feel like as the host on these things, when there's no applause afterwards, it must be quite weird and alienating after you've spent so much time speaking into the void.
Yeah, thanks for listening.
No, it was great. Thank you for the presentation. And it looks like things are going well for your project so far. I can imagine this period of almost a year now of disruption can't have been easy, particularly with having a lot of lab work. So yes, thank you so much for taking the time to present. There is one question in the Q&A so far, and if anyone is thinking of anything, please feel free to pop it in the box to ask a question. You can ask it anonymously, I think, if you prefer not to be named. So this question is asking around, it's saying, I understand Lyme disease is characterized by this gram-negative bacteria, but the peptidoglycan is strongly present in gram-positive bacteria. So why are you interested in it specifically? And then a follow-up is, why not be interested in other molecules that are more specific to this particular bacteria?
That's a great question. And there are other molecules, antigens of interest from Borrelia burgdorferi for sure. Borrelia burgdorferi actually don't have LPS, which is the lipopolysaccharide, which is highly characteristic of gram-negative bacteria. They don't have that. Their outer surface is highly decorated with other lipoproteins. And so there are other antigens of interest from Borrelia burgdorferi, certainly, and especially because peptidoglycan is not on the outside, it's not exposed, like you had mentioned. Like, opposed to gram-positive bacteria where peptidoglycan is abundant. The reason why I'm focused on this is because I introduced some of that data that showed that actually peptidoglycan was a sort of lingering component of Borrelia burgdorferi, seen in patients that were infected with Borrelia burgdorferi, that were no longer infected but still had symptoms of arthritis. And so you basically have a person with Lyme arthritis that had Lyme disease. They have this post-infectious Lyme arthritis, no evidence of Borrelia, but evidence of peptidoglycan from Borrelia. And so basically you have a person with symptoms that persist even after antibiotic treatment, and then this lingering component from the bacteria. And so we just wanted to focus, follow up on the fact that this lingering component was detected in these patients with symptoms. And so we wanted to see if that component had any immunomodulatory effects, which again, peptidoglycan is known for having immunomodulatory effects. So we just wanted to follow up that for Borrelia specifically, because like you said, I'm glad you asked that question, because intuitively you wouldn't think it would, but we're trying to see if it does.
Great, thank you. There's another question in the chat here from Alba asking about, is it known what causes the persistence? I'm assuming, Alba, please feel free to correct me, you mean the persistence of the Lyme arthritis in this proportion of patients. You explained how some people completely recover following like antibiotic treatment, but not everyone do. What can you tell us about the persistence?
And that's exactly what we're trying to figure out. Rob has come from his lab. Actually, this is his first lab, and I'm his first graduate student. And so his whole work is focused on that question: what is the persistence? How are these symptoms persisting when there isn't evidence of active infection? And so if we're asking, how do some people have persistent symptoms without active infection, and then all of a sudden we find a component from that active infection that's lingering, maybe it's that component. And so that's what we're looking into. But yeah, it's not known what's causing the persistence. Like I said, there's sort of the key piece of evidence is that these patients have a robust interferon gamma signature. And so that seems to be perpetuating their inflammatory response and thus the symptoms, but it's not known what. So the interferon gamma response is sort of the main focus. And likely a cause of their symptoms, just because interferon gamma, pro-inflammation, arthritis. But what is perpetuating that interferon gamma response is what we're trying to look into. So it's not completely known just yet.
Yeah, it got me thinking. A question I was always asked during my PhD, being based in a medical school as well, was about the sort of translational application of what I'm doing. And you've talked about the overlap with other forms of arthritis. And it got me thinking about the prevalence of this form of Lyme arthritis. Obviously, you showed us how prevalent Lyme disease is as a thing in the area of the world there. I know that it happens in Scotland too. We have ticks abundant up in the Highlands particularly. But what do you see as a translational sort of potential of what you're doing?
As far as translation goes, I think a key end goal would be vaccine development. Our lab has done some work in vaccine development, and we've worked with other labs that have funding for vaccine development as well, looking for key targets of Borrelia to use in sort of vaccine development. But as far as, I think that's sort of the general, how the general research of the lab can be applied. But as far as my project in particular, I think that what's interesting is that investigating sort of the immunomodulatory effects of peptidoglycan on arthritis in general has applications as far as any infection where or any arthritis, any form of arthritis where infection is thought to be a cause. And so yes, rheumatoid arthritis, yes, Lyme arthritis, but there has been research showing like an infection from, people with gum disease end up developing arthritis. And so there's lots of cases where arthritis is sort of a symptom of a previous infection. And if, peptidoglycan seems to be a key component of that arthritis development, that's what we're hoping to find, because peptidoglycan, like you said, is everywhere. So, and it's something that, our system is not unfamiliar with, for sure. And so that's why it's so interesting that, we find this lingering component and possible effect on the immune system when, we're used to seeing, our system is used to seeing peptidoglycan every which way.
Yeah, sure. Brilliant. Thank you very much.
Yeah,
I will now bring us on to our second speaker for this afternoon, or whatever time it is where you are today. I'm sure it's the morning for you, Joe. If I get my time difference right there, you're behind in time.
All right, let me just stop sharing here.
Great. So I'm going to be passing you over to Alba Llibre, who is joining us, I believe, from Barcelona now. I know you're from Barcelona originally, but I think now you are in Birmingham. Yes. Alba did her BA in biotechnology and biochemistry in Barcelona, and then moved to the UK to do an MSc in virology at the Imperial College London, and has had the opportunity to pursue a PhD in human immunology at Oxford, where she investigated the role of lectin-like transcript 1 in the germinal center. She's continued studying host immune responses in the context of infectious disease at the Institut Pasteur in Paris, where she focused on understanding functional immune responses to Mycobacterium tuberculosis. And now, as I said, she joins us from the University of Birmingham, having joined in March 2020, studying host immune and metabolic responses in the context of tuberculosis, funded by a Marie Sklodowska-Curie Fellowship. Over to you, Alba.
Thanks a lot, Katie. Okay, can you see my screen?
Yes, you just need to go present and it should be full.
Great.
Excellent.
So thanks a lot for the invitation and for putting this seminar series together. I've been enjoying it a lot. So as Katie just said, I moved to Birmingham a few months ago, but the work I'll be sharing with you today's work that was done at the Institut Pasteur in Paris in the laboratory of Dara Duffy. And it focuses on the identification of perturbed immune and metabolic responses during tuberculosis disease. So tuberculosis is still a global public health challenge, and is estimated that 1/4 of the worldwide population is infected with Mycobacterium tuberculosis. Now, that doesn't mean that 1 in 4 people are sick with active TB, because in about 90% of the cases, the infection remains latent and asymptomatic. And it's only at about 10% of these infected individuals that there is a progression to an active TB disease stage. And it is within the goals of the WHO by 2030 to end the TB epidemic. So we believe there's a few unmet clinical needs in the tuberculosis field that could be addressed through immune biomarkers. So the first of them is that we really need a blood-based test for active TB disease. And I'm going to devote a bit more time talking about that, because at the moment we have tests for diagnosing M.tb infection, but we don't have a blood-based test to distinguish the active from the latent stages. We need markers to identify risk for progression from this latent disease stage from the active one. We do need prognostic biomarkers for early treatment cessation, because treatment at the moment involves a 6-month course of antibiotics, which is really rough and poses several challenges. And we also need biomarkers that can help us in the development of new vaccines and treatments. So with all this in mind, our working hypothesis is that our standardized method for whole blood collection and immune stimulation could allow discrimination of active TB disease from latent M.tb infection, as well as improve general immunomonitoring in TB. So I want to present you the TruCulture system, that it's the one we use for our studies. So TruCulture is a whole blood collection and culture tube for standardized immunophenotyping procedures. So this is what the tube looks like. And these TruCulture tubes are preloaded with cell culture media and your stimulant of choice. So it could be a cytokine, it could be peptidoglycan, it could be E. coli, whatever you want. And then 1 milliliter of blood is directly drawn into the tube. And in our case, we incubated this mix of blood and median stimulation for 22 hours at 37 degrees. So with help of our collaborators in South Africa, we recruited 25 patients with active TB disease and 25 latently infected matched controls. And with their blood, we performed TruCulture stimulation with a mix of TB peptide antigens that I'm going to explain in a bit more detail, and with BCG. We also had an unstimulated control, and we kept the supernatants of these stimulations for protein analysis and metabolomics. And we also performed some flow cytometric analysis and gene expression using the NanoString platform. At this point, I'd like to say a few words about quantiferon. So quantiferon is a widely used diagnostic test for M.tb infection. So again, it's good at diagnosing M.tb infection, but not at distinguishing active from latent TB. And the way it works is that these quantiferon tubes are preloaded with a mix of 3 peptide TB antigens. The rationale behind being that if the individual that's being tested is infected with M.tb, their T cells will recognize these antigens and secrete interferon gamma, which is the readout for the assay. So we wanted to start by performing a direct comparison between the quantiferon system and the TruCulture tubes. And for this, we loaded our TruCulture tubes with the exact same peptide antigen mix that it's present in the quantiferon tube. So what we found when we stimulated whole blood from latently infected individuals and patients with active TB disease with the quantiferon tube containing these TB antigens was that all the individuals tested, except of this one here, but all of them secreted detectable levels of interferon gamma. And the levels secreted were no different between the active and latent groups. However, when we performed exactly the same stimulation with the same peptide antigens, but the only difference being that we use the TruCulture system instead of quantiferon, what we could observe is that in the active TB group, the vast majority of individuals secreted high levels of gamma, whereas only a small proportion of the latent group secreted interferon gamma. And the difference now between these two groups was highly significant. So what that means is that TruCulture is permitting the discrimination between latent infection and active TB disease in a whole blood assay, which is really nice. But these results were a bit intriguing. And what we did next was we not only looked at interferon gamma, but we measured an array of 30 different cytokines. And what we were interested in looking at was the cytokine secretion pattern in the null conditions of our two systems. So what I'm showing here is the cytokines that were secreted in the null condition of the quantiferon tube, so no TB antigen present, compared to the cytokines being secreted in the null TruCulture tube, again, no TB peptide antigen present. And what was striking to us is that in these null conditions, there were 23 proteins that were differentially secreted between the quantiferon and the TruCulture system. And that was mainly due to a non-specific activation and cytokine secretion by the innate immune cells present in the quantiferon tube. So the take-home message from this bit really is that TruCulture is a robust and reliable tool for studying immune responses. Now, one of the good things about the TruCulture system is that it's very versatile. As I said, you can really choose and play with your favorite stimulation. And as I mentioned, we looked at gamma secretion in the TB antigen stimulation, but we also wanted to look at gamma with the BCG stimulated condition. And interestingly, we could see some differences between the latent and active groups regarding gamma after BCG stimulation, but they were going in the opposite direction as in the TB antigen. So in this case, it was the latent group that secreted more interferon gamma compared to the active group. And this is interesting because we can now plot the ratio of gamma secreted within these conditions, and we can see now a very nice and clear separation between the latent infected group and the active TB group. And maybe this is nicer to show in the form of a ROC curve. So in red, you can see the quantiferon TB antigen system. So how good is quantiferon at distinguishing latent versus active TB? So we know that it's not very good at that, and therefore the area under the curve is 0.56. And we can see that when we use the TruCulture interferon gamma readout for the BCG stimulation or for the TB peptide antigens, this area under the curve, this discrimination power for the two groups increases, and we get the maximum and discriminatory potential with this when we plot the ratio of gamma secreted of BCG and TB antigens, getting 2.92, which is really good. In that respect, further work needs to be done, first of all, validating these results in a separate cohort, and also really investigating the feasibility of using something like this, you know, in a real life clinical setting. So this is all based on interferon gamma, but as I mentioned, we looked at a plethora of other cytokines, and BCG is a complex stimulation that promotes the secretion of a wide variety of cytokines. And we wanted to see which other cytokines were different between active and latent when we stimulated their blood with BCG. And interestingly, 4 of the 10 differentially secreted cytokines between TB and latent infection after BCG stimulation were members of the IL-1 family of cytokines. Now, IL-1 has been shown to be essential for control of M.tb infection. And the way it works is that IL-1 alpha or IL-1 beta binds the IL-1 receptor. Then a signaling cascade is initiated, which is mainly driven by NF-kappa beta that can translocate into the nucleus and promote the transcription of different genes, including pro-inflammatory ones. And there is this IL-1 receptor antagonist, or IL-1 array, that has the capacity to bind the receptor and preventing IL-1 alpha or IL-1 beta binding and any subsequent signaling. So we will focus in these 3 cytokines and the differences that we observed in the active and latent groups. So we saw a couple of interesting phenotypes, and I'm going to devote a bit of time to try to explain each of them in a bit more detail. So the first thing that we could observe when we stimulated whole blood from latent and active individuals with BCG is that both the protein secretion levels of IL-1 alpha and IL-1 beta were higher on the latent group compared to the active group. And the opposite trend was true for the IL-1 receptor antagonist. So in this case, it was the active group that secreted more IL-1 array than the latent group. And the other thing that was very interesting is that these IL-1 receptor antagonist differences were already present at baseline. So TB patients have higher levels of circulating IL-1 receptor antagonist compared to the latent counterparts. So focusing for a second in these IL-1 alpha and IL-1 beta differences that we could observe at the protein level, we wonder if we could observe the same differences at the transcript level, at the mRNA level. And surprisingly, for both IL-1 alpha and IL-1 beta, there were no differences at the mRNA between the latent and the active group, which suggested to us that it's probably a post-transcriptional or post-translational differences in our TB and latent groups that it's responsible for this phenotype. So looking at what we know in terms of how our IL-1 alpha and IL-1 beta produce, it's been well described in the literature now that they can be produced through both inflammasome dependent and independent mechanisms. And we wanted to check in our system which of these mechanisms was responsible for IL-1 secretion. So to test this, what we did is to stimulate whole blood from healthy individuals with BCG in the presence of either an inflammasome inhibitor or a serine protease inhibitor, which inhibits different enzymes, including granzymes that are known to play a role in IL-1 processing. And what we could observe is that both inhibitors partially prevented the release of IL-1 beta in this case, confirming that in our system, IL-1 is secreted using a combination of both inflammasome and dependent and inflammasome independent mechanisms. We then move to our gene expression data set and look at levels of expression of different granzymes. And the pattern we could observe was the same for all the granzymes, and it was also true in the null and the BCG stimulated condition. And it was that the latent group had higher expression of granzymes compared to the active group. So this is gene expression data. So we went back to these BCG stimulated supernatant samples, and we directly measured levels of granzyme protein. And what we found is that not only levels of granzyme B protein in this case were higher in the latent group compared to TB, but also that these levels of granzymes correlated very, very nicely with IL-1 secretion, suggesting that granzymes play a key role in these IL-1 differences that we observe between active and latent when we stimulate with BCG. The other key phenotype of interest that we identified was this differential circulating IL-1 receptor antagonist, the active TB group presenting more of this IL-1 receptor antagonist. So we were wondering if there were any other cytokines that follow the same pattern. And when we did this analysis, there was a very clear hit that came back, and this was IL-8. So you can see here a very nice correlation between the baseline circulating levels of IL-1RA and IL-8. So we went back again to our gene expression data set and looked for mRNA transcripts that correlated with both IL-1RA protein and IL-8 protein in TB. And we came back with a small list of genes. A substantial proportion of them were related to the PPARg gamma pathway. So PPARs are members of the nuclear receptor superfamily of ligand-inducible transcription factors, and PPARg gamma plays an essential role in metabolism and energy homeostasis, particularly in adipogenesis. Different natural ligands have been described for PPARg gamma, these including several fatty acids, as well as prostaglandins. In terms of where we can find PPARg gamma expressed in our bodies, so there's a lot of it in the adipose tissue. It's mainly present in the nucleus. This, as I said, is a transcription factor. And in terms of circulating immune cells are the monocytes who are the main expressors of PPARg gamma. It's been shown that in these monocytes, PPARg gamma inhibits production of inflammatory cytokines. It's also been shown that induction of PPARg gamma by GM-CSF is absolutely critical for differentiation of alveolar macrophages. And alveolar macrophages are the main cell target for Mycobacterium tuberculosis. And within this alveolar macrophages, PPARg gamma activation promotes an anti-inflammatory program. In the particular context of M.tb infection, it's been shown that PPARg gamma regulates both host immune and metabolic responses. And different studies have shown, on one hand, that PPARg gamma promotes CD36 expression, which is a fatty acid transporter. It also promotes IL-8 secretion, and that fits well with our findings. And there's one paper in a THP1 model that suggests that PPARg gamma activation promotes IL-1 receptor antagonist secretion. So the key question we're trying to answer here is, how do PPARg gamma and IL-1 receptor antagonists relate to each other in the context of TB in our cohort? And to test this, to investigate this a bit more, we explored our metabolomics data set, and we looked for circulating metabolites that correlated with levels of IL-1 array in the TB group. And interestingly, 5 of the 6 top metabolites that correlated with IL-1 array in TB patients were steroids of the pregnane family. So steroids that have this pregnane 4-ring structure. And just to show one example here, so between IL-1 array and one of these pregnant steroids, you can see a very nice correlation in TB patients, shown here in red, but not in the latent group. So with this in mind, we had an intense search of the literature, and we could actually find some evidence of pregnant steroids directly signaling through PPARg gamma. So the next step for us was to try to validate our findings in vitro. So for this, we designed 2 experiments. The first experiment was based on isolating monocytes from healthy individuals and stimulating them with BCG in the presence of the PPARg gamma agonist, rosiglitazone, plus or minus the PPAR antagonist GW9662, and then assess what happened with these IL-1 array levels. Experiment number 2 was a bit of the opposite approach. So we wanted to isolate these monocytes and silence PPARg gamma to then stimulate with BCG and to measure levels of IL-1 array. So what we found when we measure IL-1 array after BCG stimulation in the presence of the PPARg gamma agonist and antagonist was when we stimulated with the antagonist, that it's the dots here in black, we saw an increase in IL-1 array secretion. And this increase could be partially prevented if the PPARg gamma antagonist was present. Following the same direction, when we silence PPARg gamma in these isolated monocytes, both in the null and in the BCG stimulated condition, we could see a decrease in the levels of secreted IL-1 array. These all pointing towards the fact that signaling through PPARg gamma results in IL-1 array secretion. So at the moment, our working model is, we imagine these pregnant steroids being able to get into the cell, binding PPARg gamma, which then translocate into the nucleus and initiate the transcription of several genes, including CD36, that we know is important for lipid uptake and therefore for M.tb growth. But it also, we suggest it would also contribute to IL-1 array transcription, translation, and secretion that would negatively impact IL-1 signaling and would favor uncontrolled M.tb infection. So to conclude, TruCulture provides a robust and reliable immunomonitoring tool for analysis of functional immune responses in TB disease. In particular, I've shown you today how TB antigen stimulation allows discrimination between latent and active TB based on this interferon gamma readout. Also, we know that BCG stimulation revealed a dysregulated IL-1 pathway in active TB with a key role for granzymes. And finally, that baseline high concentrations of the IL-1 receptor antagonist in tuberculosis patients are a result of increased PPARg gamma signaling through pregnant steroids. And with this, I'd like to finish by thanking everyone that's been involved in this work, particularly Dara Duffy's group at the Institut Pasteur in Paris and Tom Scriva's team in South Africa. And thank you all for your attention.
Thank you. You can see that the sun has set here. I've rapidly lost daylight in the last sort of 45 minutes. So I apologize that I'm now joining you in the dark. It's the hazard of living so far north. But thank you so much for your presentation. That was really, really, really interesting. I will confess to not being an immunologist by background, so I don't have a lot of sort of basic knowledge, but one of the things I recognized was the term BCG, because that's what they refer to the vaccine as in the UK anyway. What exactly is BCG in the context that you're using it?
So BCG is a bacteria of the same family, very, very similar, but infects animals instead.
Right.
It's very similar and it confers good cross protection. So we use it as a live inactivated vaccine for TB. But as you know, there's been a lot of talk in the context of COVID as well, and whether, you know, countries that have had BCG vaccination for a long time seem to be maybe cross-protected against other kind of infections. That's being dismantled at the moment, but yeah.
Yeah. I wondered around the importance of distinguishing between live and like active TB infection and the latent infection. How does that apply in a sort of public health setting in terms of like managing the spread of TB? Because obviously, I'm assuming if you have latent TB, you can still pass it on.
So what we think at the moment is that you can mainly pass it on when you are active. Right. And the thing as well is that because 1 in 4 people in the world have latent TB, it's just not feasible to treat, you know, 25% of the world population. So the key thing would be able to identify the latently infected individuals that are kind of on the way to progression to active TB disease, to be able to treat those. So from a public health perspective, it is very important because, again, we can treat 1 in 4 people in the world, or it's very challenging, at least. Yeah, absolutely. Do you see this trajectory of this goal of you know, is it elimination by 2030? Is that the plan? Yeah, eradication. I mean, because the WHO publishes like a yearly report, and it looks like we're pretty much behind on every possible thing we can measure to what the aim should be to reach that goal by 2030. COVID as well has had a massive impact, and people are really worried on how that will impact, you know, TB cases in the coming years. So yeah, it's an ambitious goal, and at the moment it doesn't look extremely promising, unfortunately. On the question of COVID and the impact on TB cases, has there been evidence to suggest that it's decreased or increased? So what's very clear is that they were really effective. So TB is mainly endemic in southern sub-Saharan Africa and southeastern Asia. Yeah. And there were really effective programs put in place to, you know, to diagnose TB, to make sure that everyone that gets diagnosed can get treated. And all these programs have been severely impacted by COVID, by people's inability to be able to travel to places, or people being scared of, you know, going out and about. So people are really scared that there will be like a bit of a rebound in that respect, but who knows? That's really interesting. I hadn't thought about that angle of it so much. The reason I ask about spread is because I work with colleagues now who support people with HIV/AIDS in Scotland, and in the UK during COVID they've reported the lowest rates of infection from HIV/AIDS, and they think largely that is because people are being so much safer and not having contact with other people. But that's obviously in the
the global west and north.
I mean, that's also possible that, you know, the new cases maybe have decreased, like, because, you know, if everyone stayed at home, there is less room for, but I think they were particularly worried about, you know, because it's a 6-month antibiotic treatment and compliance is already complicated. So if on top of that, you have so many restrictions on, you know, traveling and moving around. Yeah, I don't know.
Yeah. Thank you. And thank you again to Joe for both of your presentations. It was really brilliant and lovely to meet you both today. And I don't think there's any other follow-up questions in the chat, but as ever, this recording should be made available soon via the STEM Village YouTube channel. So people can always catch up if they've missed anything and get in touch with the panelists. Thank you so much and have a nice evening or rest of your day, depending on where you are in the world. Thanks a lot.
Thanks a lot.
Thank you.